Biology, Chapter 11, Section 3, Chromosomes and Human Heredity. Our main idea for our final section for this chapter, chromosomes can be studied using karyotypes. Your 4 essential questions are, how are karyotypes used to study genetic disorders? What is the role of telomeres? How is non-disjunction related to Down syndrome and other abnormal chromosomal numbers? What are the benefits and risks of diagnostic fetal testing? So first, karyotype studies. Scientists study not only genes but entire chromosomes. Images of chromosomes stained during metaphase allow scientists to study sister chromatids and homologous chromosomes. A karyotype is a micrograph or a picture in which the pairs of homologous chromosomes are arranged in decreasing size.
A karyotype shows the size, shape, and number of chromosomes in an individual organism, and the karyotype shown here is of a normal human male. So what scientists do to create a karyotype, they'll take cells from the individual, grow them in a lab, and then freeze them at metaphase of mitosis, because this is the phase when DNA is fully condensed into chromosome form. You can see in this human karyotype that we have 23 pairs of chromosomes. 22 of which are autosomes, which just means they aren't sex chromosomes, and then 1 pair
This is your pair of sex
right
chromosomes
here.
right here.
So you can tell this is a male human because there is an X and Y chromosome present. This is the X, this is the Y, and notice how the Y is a lot smaller. In females, there would be 2 X chromosomes present. These karyotypes can be important tools for analyzing and researching an individual's genetic makeup, particularly for identifying genetic diseases. For example, Down syndrome is caused by having a partial or full 21st chromosome. So it would look something like this. You'd have another chromosome right there. And a karyotype quickly helps identify this condition. A pregnant mother can also choose to have a karyotype done on her developing baby to look for potential genetic disorders.
The protective caps on the end of chromosomes are called telomeres. They consist of DNA and proteins. They serve as a protective function for the structure of the chromosome. Non-disjunction is when cell division, where sister chromatids fail to separate properly. Okay, so we have sister chromatids, as you can see in this picture here. And when they separated, it didn't happen the way it's supposed to be. One of these strands is supposed to be in this circle, and the other circle is supposed to have just 1 strand. So when the sperm comes into play and it fertilizes, one of its DNA goes over here, which is red, and the other one goes here. So as you can see from the color coding, make sure when you color it, it looks like that. The egg or the female is blue. The chromosomes in, the sister chromatids in the sperm are red. And so when we get down here, we have monosomy, which is just 1, not enough. And over here we have 2 of the female's contribution and only 1 of the males. So Down syndrome is a result of an extra chromosome 21. Characteristics include distinctive facial features, short stature, heart defects, and mental disability.
Karyotypes can often reveal an incorrect number of chromosomes. This condition is known as aneuploidy or non-disjunction. Non-disjunction is the process that produces the incorrect number of chromosomes. And this results from an error, usually during meiosis, in the formation of the egg or the sperm cells. An example that you are probably all familiar with is Down syndrome. Down syndrome is also known as trisomy 21. Tri-21. So rather than being 2 copies of chromosome 21, 1 from the mother and 1 from the father, there are 3, as we can see down here.
Some of the symptoms of Mitosis syndrome include a small head, smaller missing eyes, heart defects, extra fingers, abnormal genitalia. They can be mentally retarded, having a cleft palate, which is seen in this picture here. Most of them do die within a few weeks after birth. This is going to have an extra chromosome number 21. So again, this is due to what's called a non-disjunction event. So those chromosomes are all lined up, ready to separate, half into one of the eggs half into another one of the eggs, and for some reason number 21 doesn't separate correctly, and either in that sperm there's 3 of number 21, or in that egg there's 3 of number 21. Okay, you can see all the other homologous chromosomes are set up and match nicely. Some of the symptoms of Down syndrome include short, broad hands, stubby fingers, rough skin, impotency in males, mentally retarded, having a small round face, protruding tongue, and they do typically have a shorter lifespan into their 30s.
And because I know you need it, let's take a look at a simulation between mitosis and meiosis. Yes, watch it a few times because there will be quizzes all throughout the year, and it's on your final as well as your EOC.
Okay, today I'm going to model mitosis, and we're going to do it with a simple organism. In this organism, it's going to have, we'll make it 4 chromosomes, and I'll add those other 2 in just a second. And so our diploid cell is going to have a diploid number of 4. What does diploid mean, haploid mean? Well, in this case, diploid means that you have a chromosome from your father, that you get from your father, and a chromosome that you get from your mother. Now, we would call this chromosome 1, and that's because it's the longest one. If we had 2 other chromosomes, this would be chromosome 1, and this would be chromosome 2. Now in us, we have 22 what are called autosomes, and then we have sex chromosomes. But the way chromosomes are named are just their number. 1 is the longest, 2 is the next longest, 3 is the next longest. So a few other things you should know about a chromosome. This would be the centromere, the center where they're connected, and then each of these beads represents a gene. Now here we just have a few genes, but in a human cell, we're going to have thousands of genes on each of the chromosomes. And so this is a very simplified model. You can imagine if this is a human chromosome, my whole board would be filled with these chromosomes. And so this is a very simple model. So this one right here from Dad and this one from Mom are what are called homologous chromosomes. And what that means is that they have the same length, but they don't necessarily do the same thing. What does that mean? Chromosome 1 from Dad and 1 from Mom are going to be exactly the same length, centromere is located in the same thing, and the genes will be in the same spot. So for example, if this is the gene for blue eyes, this would be where the other gene for blue eyes is. And so you may be asking yourself, well, how are they different then if everything seems to be the same? Well, this right here could be a recessive gene for blue eyes, and this could be a dominant gene. It doesn't give you blue eyes. And so those alleles are going to be found on either side. Or this right here could be the gene for hitchhiker's thumb, makes your thumb bend backwards like this. Then over here, so hitchhiker's thumb here, this could be one for a straight thumb, so you don't have a hitchhiker's thumb. And so the chromosome you get from Mom, Dad, and the chromosome you get from Mom are homologous, and they never ever meet, except in meiosis, which we'll get to in just a second. Okay, so what's our goal in mitosis? The goal in mitosis is to make an exact copy of the nucleus. And so this circle right here, bounded by this, is going to be the nuclei, and these are going to be the chromosomes inside it. Now, what does a cell do before it divides? A cell will get bigger, and then it will copy its DNA. So let me model how that works. When it copies the DNA, this chromosome will have an exact copy of it made. This occurs during the S phase. This chromosome will have an exact copy of itself. This one will have an exact copy of itself. And then this one will make an exact copy of itself. And so maybe this is what you remember chromosomes looking like. And so after the S phase, you'll eventually have a cell or a chromosome that's made an exact duplicate of itself. And so if this is a gene here, there's an exact copy on this side. In other words, this side and this side are exactly the same. In fact, those are called sister chromatids at this point. And so this would be at the end of the S phase. What happens after the S phase? It goes into another growth phase called G2 phase. So G2 phase, all of that is part of interphase. Now, would it look like this during interphase? You wouldn't see the chromosomes during interphase. All of that DNA would be loose within the cell, doing its job, the job that it normally does. And so you're really not going to see chromosomes look like this until we get to prophase. Okay, what happens during prophase? All that DNA is going to coalesce and we'll actually be able to see it. Okay, what happens next? So that would be prophase. The next thing is going to be metaphase. What happens in metaphase is that these will little spindle fibers will attach to these, and they will line up and meet in the middle. And so they're going to line up like this. This chromosome will line up like that, and this one will line up like that, and this one will line up like that. And so the reason they line up like that is that the spindle fibers attached to them, and they're going to go from here to here, and here to here. And then we're going to have attachments over on the other side as well. Now these spindles are going to go outside of the nuclei, which I would draw, but I can't quite fit that on my screen right here. And so there's going to be a tug on either side by the spindles, and that lines these all up in this perfect, what is called the metaphase plate that goes right down the middle. So this would be metaphase. What happens to next is going to be anaphase. And so in anaphase what happens is these are going to be pulled to the side as those spindles shorten. They'll be pulled like this. And these will be pulled. This all happens at the same time. And these will be pulled like this as well. So they're going to move to the sides. And then the spindle fibers are going to start to disappear. And then we're going to have two brand new nuclei. So this would be one nuclei, and this would be another nuclei. Now if you look at the chromosomes in each of these nuclei, we've got one like that, we've got one like that, we've got one like that, we've got one like that. They're exactly the same. In other words, we have two brand new nuclei, and each of them have the same duplicate DNA in each one. And that's the goal of mitosis. The goal of mitosis is to make two exact copies of the cell. Okay, so I'm going to take a minute and clean this up for just a second, and then I'm going to show you how that differs from meiosis. So in mitosis, we're making two nuclei, and those nuclei are going to be exactly the same. But now let me show you what we do in meiosis. In meiosis, our goal is not to make duplicate cells, but to actually make different cells. We make genetically different cells. And so how does it start? Well, it starts the same way. The cell's going to make a copy of itself. And so it's going to go through a growth phase, so the cell will get larger. It's then going to duplicate its DNA, so it's going to duplicate its DNA like that, and like that, and like that, and like that. So this is going to be at the end of interphase. So the cell has grown, it's copied its DNA, and then it started to grow again. But now we're going to have something different happen. And so in meiosis, we have two divisions. And so during the first division, what will happen is the homologs will come together. And this forms something called a tetrad. And so we'll have this homolog together and this homolog together. In other words, the chromosome that you get from your dad and the chromosome that you get from your mom will actually come together, and they'll wrap around each other like this. They line up along the middle, so this would be in metaphase 1. And they are going to, this actually happens a little before this during prophase, but what will happen is that they're going to wrap around each other so closely that portions of one will switch with portions of another. And portions of this one will switch with portions of another one. And so during prophase 1, they're going to switch bits of them. Or this one right here, it might pop off here, this little gene, and it might switch with another gene over here. So it'll switch like that. And what that gives us is variability. And so this would be during prophase 1, we cross over, and so we switch some of the chromosomes. Another important thing happens at the next step, which is metaphase 1. This might line up like this, but it also might line up like this. And this might line up like this, but it also might line up like this. And this is called independent assortment, as I start to lose one of my chromosomes here. In other words, depending on which side they line up on, they're going to be pulled in a different direction. Now, why is this important? Well, let's say this right here is the gene for Huntington's disease. Huntington's disease means you're going to die when you get to be 40 years old. And if it lines up like this and goes to a sperm or an egg over here, then you're going to get Huntington's. But if it goes over here, you're not. And so this, when you draw a Punnett square, is where that actual genetic 50-50 split takes place. That's called independent assortment. Okay, so now what happens next? We're going to attach a spindle on each of these. So there's going to be a spindle here, and there's going to be a spindle here. And that spindle is going to, during anaphase 1, is going to pull them apart. So these are going to pull apart, and these are going to pull apart to the side. Now what happens next is we form two new nuclei. So I'll do that like this. So we form 2 new nuclei. And now the next thing we go through is we don't go through another interphase. And so there's